Dear students, research scholars, members of the faculty, eminent academician, a warm greetings to you. I am J. Dinesh Raja, serving as an assistant professor in the Department of Biotechnology and Bioinformatics in Bishop Heba College, Tiruchirappalli, Tamil Nadu. Today, we are going to discuss about the widely using technology in the field of plant biotechnology is called plant tissue culture. Plant tissue culture is a technique that we are using in the field of agricultural, forestry and industrial biotechnology. What is plant tissue culture? Plant tissue culture is a technique of growing plant in a in vitro condition. Mostly endemic and endangered species of plants can be used for plant tissue culture. Plant tissue culture is a collection of techniques used to grow the plants in a artificially prepared condition with the help of suitable temperature, suitable medium and the suitable hormones. Without the plant tissue culture, the plant that are going to be in an endangered or the plants that are going to be in an endemic condition cannot be retrieved. With the help of this plant tissue culture, we can use or we can produce more number of plants by using a single explants. With the help of plant tissue culture, we can create transgenic plants. We can produce more number of orchid plants. The plants which are medicinally important, the plants which are widely used for the purpose of producing medicines can also be retrieved with the help of plant tissue culture. Worldwide, plant tissue culture technique is used in all fields, especially in the field of agricultural, medical biotechnology, forestry and industrial biotechnology. Plant vaccines can also be produced by using plant tissue culture technology. If you want to know about the plant tissue culture technique, please kindly go through the PowerPoint presentation. Plant tissue culture. Plant tissue culture is a collection of techniques used to maintain or grow plant cells, tissues or organs under sterile conditions on a nutrient culture medium of known composition. Plant tissue culture is widely used to produce clones of a plant in a method known as tissue culture. Advantages of plant tissue culture. The production of extract copies of plants that are produced, particularly good flowers and fruits, to quickly produce matured plants. The production of multiples of plants in the absence of seeds. The production of plants in a sterile containers that allows them to be moved with greatly reduced chances of transmitting diseases, pests and pathogens. The production of plants from seeds that otherwise have a very low chances of germination and growing, that is orchids. To clear particular plants of viral and other plants can be produced. What are the advantages of plant tissue culture? Availability of a raw material, variation in supplies and quality, patent rights, easy purification of the compounds, modification of chemical structure. Explant. Any part of the plant taken out and grown in the tube under sterile condition, especially nutrient media, is called explant. Methods of plant tissue culture. Plant tissue culture includes two major methods. These are called a type of in vitro growth callus and suspension culture, type of an explant single cell culture, shoot and root cultures, somatic embryo culture, meristem culture, anther culture and haploid culture. And this slide we are going to learn about the environmental conditions. There are three important aspects in vitro culture: nutrient medium, aseptic condition, aeration of the tissue. Nutrient medium. Medium depends upon the type of plant tissue culture or cell used for culture. Generally, nutrient consists of inorganic salts, both macro and micro elements, a carbon source, usually sucrose, vitamins, example nicotinic acid, thiamine, and pyridoxine. What is called aseptic condition? Nutrient medium contains sugar, which increases the growth of microbes. These microbes compete with growing tissues and finally kill it. It is important to maintain aseptic condition. Sterilization is very important step to stop the growth of unwanted microbes. Aeration of the tissue. Proper aeration of the culture tissue is also an important aspect of culture technique. It is achieved by occasionally stirring the medium by stirring by automatic shaker. And this slide we are going to learn about the types of plant tissue culture. Callus culture. Callus culture, cell division in explants from callus. Callus is an irregular, unorganized and undifferentiated mass of actively dividing cells. Darkness and solid medium gelled by agar stimulated by callus formation. The medium contains dioxins and BAP, that is benzylaminopurin. Both are growth regulators, hormones. Maintenance of a callus culture. After sufficient time of callus growth on the same medium, following changes will occur. These are depletion of a nutrient in the medium, gradual loss of water, accumulation of metabolic toxins. Hence, for the maintenance of growth in callus is it. Single cell culture. As stated earlier, cells divided from a single cell through mitosis constitute a clone, and the process of obtaining clones is called cloning. That is, asexual progeny of a single individual make up a clone. There are two popular techniques of a single cell culture. Root tip culture. Tips of the lateral roots are sterilized, excised and transferred to the fresh medium. The lateral roots continue to grow and provide several roots. After 7 days, that are used to initiate stock or experiment. Thus the root material derived from a single radical. Leaves culture. Leaves may be detached from shoots, surface sterilized and placed in a healthy condition for a long period. Growth rate in the culture depends on their stages of maturity at excision. Young leaves have more growth potential than normal leaves. Shoot tip culture. The excised shoot tip culture of many plant species can be cultured on relatively simple nutrient media. This media must contain growth hormones and will often from roots to develop into whole plants. Complete flower culture. Flowers are excised, sterilized by immersion of 5% of calcium hypochlorite, washed with sterile water, transferred to this culture tube containing an agar medium. Fruits which develop are smaller than their natural counterpart. Size can be increased by normal flower culture. Anther culture. Young flower buds are removed from the plant and surface sterilized. The anthers are then excised and transferred to an appropriate nutrient medium. The plants are formed after 4 to 5 weeks of inoculation. Pollen culture. Pollen grains are removed from the anther. Anthers are placed in a 5 ml liquid medium in a petri dish. A petri dishes containing the pollen grains in the culture media are sealed with a parafilm and incubated at 28 degrees Celsius in dark for 14 days. Application of plant tissue culture. Tissue culture is used to conserve the rare species in the forest. A plant breeder may be used to tissue culture to screen cells rather than the plants. Large scale of growth of plant cells in liquid culture, a source of secondary products like recombinant protein used as a factors. To cross distantly related species by protoplast fusion. Help in crop improvement. Plant tissue culture creation of additional genetic variation. Plant tissue culture used in selection of plants resistant to toxins, viruses, etc. Suitable for breeding for tree species. Tissue culture have an advantage of time saver. With the help of root culture, plants are ready to grow, which save time. Next, we are going to learn about the polyploid. Polyploidy refers to when an organism has more than 2 complete sets of chromosomes. This condition is frequently found in plants. Polyploids can be divided into types based on the number of chromosomes they carry. One is triploid, second one is tetraploid, third one is pentaploids. Importance of polyploids: identify the genetic origin of the crops, generate new plant genotypes and species, enable interrogation of genes from related species, self-fertilization. A plant may be made sterile by developing a triploid fertile tetraploids and diploids or hybridized to get triploids. Polyploidy can be used to obtain seedless fruits such as the seedless watermelon. Another example of successful polyploidy is the banana, which are triploid, seedless and sterile. As with the hybrids, polyploidy have been observed to be a bigger and more robust than their diploid parents. This may be include bigger leaves, flower and fruits as well as increased height. An example of the strawberry plant diploid varieties of seedless watermelon and strawberry. Mutation. Mutation is the inheritable changes in the characters of an organism. Mutation can occur spontaneously in nature or they may be induced experimentally. Mutation can be beneficial or harmful to an individual. Importance of mutation. Evolution of new and better food plants having good desirable characters such as higher yield and greater resistance. Protection of a new species. Agricultural revolution. Mutation breeding often provide solution for some specific plant breeding problems. Improvement in genetic characteristics so that the new species give desirable effects to increase crop yield. And finally, we are going to learn about hybridization. Hybridization is the process of crossing two genetically different individuals to result in a third individual with a different one. Dear students, research scholars, members of the faculty and academician, this is the technique of plant tissue culture and these are all the applications of plant tissue culture. Hope that you have experienced the value of plant tissue culture and try to use the tissue culture facility in your institution and in your academic research. All the best. Thank you very much for your patient listening. Thank you and all.